that. Okay. So couple updates. I've updated the gradebook, so you should see your test scores now. I'm having a meeting with the other professors about how the exam went, so there may be an additional update about that, not totally sure. I've also put in the anatomy and physiology lab grades for the things that everyone has completed. So anything that was due this week isn't in yet. I did notice that some of you are missing like a bunch of physiology grades. So if that's you, I don't know anything about phys lab this semester, but a thing to check in on because I don't want you guys missing those points. Also, mastering A&P stuff should be updated now. Okay. Beyond that, we're gonna continue on with the gastrointestinal system and maybe move into the reproductive system. I'm sure you're all excited. This is the last week of classes. All right, almost through. Okay, so when we left off, we were talking about control of the gastrointestinal system, mainly control of things like secretion. We hadn't gotten into motility yet, and we'll talk about that in a second. Okay, so overall, we had talked about basically how different parts of the nervous system control the gastrointestinal system, and how some of the stimuli that we're interested in come from the head, some of the stimuli come from the stomach, some of the stimuli come from the intestines. And we're gonna think more about that today. We also had this idea of short reflexes versus long reflexes, which just has to do with the fact that sometimes these stimuli are going to send signals up to the brain. Sometimes these signals are just directly kind of staying in your GI tract, staying in the abdomen. Okay. So we talked about saliva secretion and acid and pepsinogen secretion on, what day is today? We talked about it on Friday. Today is Monday. Okay. So we talked about saliva secretion, acid and pepsinogen, and how we're secreting acid into the stomach, really when we have food in there to digest. Okay. So we talked about the cephalic phase regulation of secretion, really is where we left off. So we talked about the fact that any indication of the presence of food really is going to stimulate the secretion of acid and pepsinogen. And remember that like acid and pepsinogen are kind of linked because we need to use the acid to activate pepsinogen, turn it into pepsin so that we can break down proteins. So if we see food, if we taste food, if we smell food, if we chew food, if we swallow food, that's all external to your abdomen. It's like above your stomach. So that counts as cephalic phase regulation. Those signals are really going to go to your brain, to the center that controls digestion, and then those nerves are going to send signals way back down, basically to your stomach. So all of these are really activating our parasympathetic nervous system, which is one of the pieces of our autonomic nervous system. The parasympathetics are the rest and digest part. So we looked at a diagram of what this would kind of look like if we mapped it out. So if we saw, smell, taste, chew food, we increase parasympathetic activity from the brain. So some of that is going to just directly increase acid and pepsinogen secretion in the stomach. And we also have sort of an endocrine pathway where we're going to have parasympathetic activity increasing the secretion of gastrin, which is like a hormone. So it's a hormone, so it goes into your bloodstream, so it's in the plasma. But then that gastrin in the plasma also increases acid and pepsinogen secretion. So these are like two pathways that are basically amplifying each other. Okay. So new stuff, yeah, is the gastric phase regulation. Okay. So when we're talking about gastric phase regulation, we're thinking about stimuli directly in the stomach. So basically, this is just that we're going to trigger the release of acid pepsinogen, and our relevant hormone gastrin when your stomach senses that there's stuff in it to digest. So there are basically two ways it can sense that. One is that it can actually sense the proteins, peptides, and amino acids themselves. So it's going to be, like chemically sensing proteins and the things that you get from proteins when you start digesting them. Or we can physically sense that our stomach is getting bigger, that we have distension, that those rugae, those kind of folds in the stomach, are flattening out as your stomach gets bigger. And that's also going to inspire your stomach to start secreting acid, pepsinogen, and gastrin. So are things that are going to help us increase our digestion, that are going to help us break down that food. So if your stomach can tell that there's food in it, it's going to produce stuff to digest that food. By the time we get to gastric phase regulation, we already do have some acid and pepsinogen in there to start digesting the food. That's why this says like proteins, peptides, and amino acids, right? Because when we saw the food, when we swallowed the food, we had that cephalic phase regulation, kind of prepping our stomach for it. So this is amplifying something that's kind of already going on. Okay. So we have another flow chart. So here we see on our flow chart, basically what's happening from the chemoreceptors and the mechanoreceptors. And those are just like receptors that sense either the proteins or the stomach distension. So here in this diagram, we see our initial stimulus. So the thing we're reacting to is either proteins and protein digestion products. So like our broken up polypeptides, all the way down to amino acids. So those would be sensed by the chemoreceptors. So that's this part in the first half of this part in yellow, this first part in yellow. Or the other thing we could be sensing is the distension of the stomach. So your stomach getting bigger, the rugae flattening out. And that would be sensed by mechanoreceptors. So they're physically kind of sensing like stretch of the stomach. So they're detecting and responding to this. We're not going to focus on exactly which nerves they're triggering. So they're in the next white box. We just have that there are short reflexes and long reflexes. So the chemoreceptors and the mechanoreceptors are going to send signals up to the brain, to the CNS, to your processing centers. And they're also going to have little short nerves that just stay in your abdomen. They're going to basically control the secretion through both of those pathways. So just like in the previous diagram, we have like, we have two things that are happening here after we have these chemoreceptors and mechanoreceptors responding. So one thing is that we have our reflexes going directly to the parietal and chief cells, okay? So this is probably, right, a short reflex because it's just staying kind of here in the stomach. So our parietal cells and our chief cells, remember, are those specialized cells for secretion down in the bottom of the gastric pits. So they stimulate the secretion of acid and pepsinogen. That's where we had those cells, right? That's where we have those cells secreting those things. Okay. So our chemoreceptors and mechanoreceptors have that as one kind of direct effect. And then we have a side branch of this diagram. So our side branch here is that we have a sort of more circuitous pathway that ultimately is going to do the same thing. It's also going to increase acid and pepsinogen secretion, and therefore acid and pepsinogen available in the stomach to break down our food. And on this more circuitous pathway, we have these short and long reflexes stimulating the G cells. The G cells are also in our stomach, but they have an endocrine function. So endocrine means that we're putting a hormone in the bloodstream in order to create some type of reaction. So our G cells are increasing the secretion of gastrin, which is our relevant hormone. So they're putting that gastrin into the plasma. So the gastrin goes into your blood. And then gastrin in the plasma also affects the parietal and chief cells. So again, this is just increasing the acid and pepsinogen secretion. Okay. So as your food continues moving through your GI tract, you don't want to like permanently up the acid in your stomach, because eventually that food is going to pass into the duodenum, into the jejunum, into the ileum, and then into your large intestine. So we also want to turn off the secretion of our acid, pepsinogen, and gastrin, because gastrin is like upregulating the secretion of these two products. So there are basically two ways that we're going to inhibit the secretion. One is that we can sense the food leaving the stomach. So when we sense food leaving the stomach, I'm going to flip back for a second, that's like going to turn this off, right? So this diagram was how we turned on our acid and pepsinogen secretion. We can also turn it off. So if we were to turn it off, we no longer have proteins in the stomach. We don't have distension of the stomach anymore. So our chemoreceptors and mechanoreceptors are like going to stop firing. So as they stop firing, this means they're not going to be stimulating acid and pepsinogen secretion anymore. So we're going to be stimulating less of this. Okay. So that's turning off this signal. And as they're not sending signals out anymore, because they're not sensing this anymore, they're also going to stop secreting gastrin, which means we're going to get less and less gastrin in the plasma, which means it's going to stop inspiring us to increase acid and pepsinogen secretion as well. So once we don't have this step, once those chemoreceptors and mechanoreceptors are no longer detecting and therefore no longer responding, these pathways turn off. Okay. So another thing that's happening as food is leaving the stomach. So when food leaves the stomach, this means there's nothing really anymore for the acid and the pepsin that we've turned on to digest, which basically means that we're going to have a bunch of hydrogen ions kind of collecting in the stomach with nothing really to do. And so we're going to see increasing acidity in the stomach. So as we have increasing acidity in the stomach, we don't have a chart for this, but I'm going to scroll back and basically show you where this would be happening. So like looking at this diagram, if instead of chemoreceptors and mechanoreceptors here, let's say we have a detection of increasing acidity in the stomach, it's going to basically affect this pathway here with the G cells. So as we have receptors sensing increasing acidity, that's going to inhibit gastrin secretion. So that arrow would be pointing down, be inhibiting gastrin secretion, which means that we'd be turning off this right-hand side of the diagram in a different way. So we'd be inhibiting gastrin secretion in response to high acidity, which means we'd be decreasing plasma gastrin, which means we'd be decreasing acid and pepsinogen secretion. So that would all be gastric phase inhibition of the secretion. So we're like turning off the signal coming from the stomach. The other thing that we have happening is that as food is passing from the stomach, it's like going somewhere. The place it's going is into your small intestine, and specifically into the duodenum. We're going to see a bunch of things happening in the duodenum. We're going to see that the entry of food into the duodenum first inhibits acid and pepsinogen secretion, and then it's also going to increase secretion of some other hormones that are going to affect our pancreas, basically, our pancreas and our liver. So we're going to want enzymes going into the duodenum instead of into the stomach. That makes sense because that's where the food is now. So things that happen when food gets in the duodenum, right? So as we're contracting our stomach, we're moving chyme into the duodenum past our sphincter, right? We have a sphincter between the stomach and the duodenum. So our pyloric sphincter, opening and closing, slowly moving chyme into our small intestine. So now we have like a bunch of amino acids in there. We have, some maybe like glucose. We have some components of carbohydrates. We've got little pieces of molecules in our duodenum. So those pieces of molecules, they are not water. And therefore, they're increasing the osmolarity of the fluid in the duodenum. So before we just had whatever is left in the duodenum, right? Like it's kind of empty, doesn't have much in it, so it has relatively low osmolarity. Now it has a bunch of pieces of proteins in it, so it has increased osmolarity. So that's one thing that can be sensed basically by receptors in your intestines. Can also sense the fact that there's increased fat in there. So we know we have lipids coming from the stomach. They're moving into the duodenum, which is the main place that they're going to get processed. So that's where they're going to have those lipases breaking them down. That's where they're mixing with bile salts. So one of the things we're sensing in the duodenum is fat. As we're moving this chyme in, we're also moving acid from the stomach to the duodenum. So the acid was produced in the stomach. It was mixed up with our proteins, our carbohydrates, our lipids, whatever we ate. We squeeze that mixture into the duodenum. It means we're squeezing the hydrogen ions into the duodenum as well. So we can sense that the duodenum is getting more acidic because the stomach acids are moving in. Finally, we can just sense physical stretching of the duodenum. So just like in the stomach, we can sense that the stomach is getting bigger. We could also sense that the duodenum is getting fuller, getting bigger. We got these stretchy walls, right? So we can sense distension in the duodenum. So there are going to be long and short reflex pathways. So nerves that are staying within the abdomen, nerves that are going back to the CNS. Both of these pathways are going to inhibit acid and pepsinogen secretion when they sense that your food is basically moved on. Okay. So in addition to like turning off our stomach, we now want to turn on the duodenum, right? So the point of the duodenum, like what's happening there, is we're doing two things. We're continuing breaking down all our nutrients, right? So we're continuing to break down our proteins with our new enzymes for protein digestion, right? We've released zymogens into the duodenum and we're activating them. We have our lipases that we're secreting into the duodenum, where we want to use those lipases to break up our fats, or sorry, we want to use bile salts to break up our fats. We want to use the lipases to digest the fats. And the other thing that we want to do in the duodenum is we're putting in the chyme from the stomach. It's really acidic, right? So we can sense the acidity in the duodenum. We want to counteract that acidity. You don't want all this matter going through your intestines to stay really acidic. The acidity has done its job in the stomach. It's done what we want it to do, which is basically like denature proteins, so unfold them. So the other thing that we're going to be trying to do in the duodenum is to buffer out those hydrogen ions, which means that we're going to pump some bicarbonate into there. So just like we buffer pH in the plasma with bicarbonate, we're doing that in the duodenum as well. And the place that the enzymes to continue digestion in the duodenum are coming from is the pancreas. Place that the bicarb is coming from, also mainly the pancreas. So we're getting the enzymes and the bicarb from two different parts, basically, like two different types of cells, the acinar cells and the duct cells. So the pancreas is a gland, right? So it has these acinar cells, which are like little bubbles, right? So like little bubbles, kind of like we remember we have alveoli in the lungs. We got acinar cells, little bubbles in our glands, specifically in the pancreas. We also have ducts. So ducts, right, are just tubes leading from our gland into wherever it's going. In this case, leading from our pancreas into our duodenum. And those duct cells are also going to produce components of the juices that are going into the duodenum. So in our acinar cells, what we're producing are enzymes mainly. So we got, it's pretty watery, basically, the fluid that the acinar cells are making. So it's got water, it's got some ions, and the digestive enzymes. That's a relatively small volume of what's coming out of the pancreas, we have a lot of stuff coming out of the duct cells. So we're making the enzymes and these little bubbles at the ends of our ducts, right, our acinar cells, and then they're putting that fluid into the ducts. And then as that fluid continues, the duct cells are making more fluid, which is mainly like the main functional component of that stuff made by the duct cells is bicarbonate. Okay, so we're gonna take a look at how the acinar cells and those duct cells are stimulated to make these things. So our two stimulants are CCK and secretin. So our acinar cells and our duct cells are basically regulated independently of each other. Usually when we're turning on our duct cells or turning on our acinar cells, we're gonna turn on both of them together, but they each have their own pathways. Okay, so this is basically because pancreatic juice isn't always the same. So sometimes you're going to have like a lot of protein in your duodenum. So you're going to want to make a bunch of enzymes to digest that protein, but sometimes you're not, right? Like fluid that you drink also goes into your stomach, goes into your duodenum. And in that case, you might not need to break down as much. You might not need to break down as much protein. So we want to be able to control our enzymes and our bicarbonate secretion separately. So first thing first, the first thing that we're going to stimulate is basically for breaking stuff down. So we're going to be secreting enzymes from the acinar cells. And what turns on the acinar cells is CCK. So CCK's main job is to tell those little bubbles at the end of the ducts, the acinar cells, to secrete enzymes to continue digestion, to break down food. CCK does have a secondary role, which is that it stimulates bicarbonate release a little bit. Actually, I'm going to come back to that second point because these things kind of work together. Okay, so CCK's main job is to stimulate the acinar cells and their secreting enzymes. The other thing that we need to turn on is secretin, because secretin stimulates duct cells to secrete bicarbonate. So if you're going to associate, like word associate one of these stimulants with one of these products in the pancreatic juice, CCK goes to enzymes, secretin goes to bicarbonate. So this is true, but these systems do work together. So in addition to causing the secretion of enzymes, CCK is a little bit of a stimulant for bicarbonate release. It doesn't do much, but it does a little bit. And the same is true for secretin. So secretin mainly stimulates duct cells to secrete bicarbonate, because that's where bicarbonate is coming from. It's also a little bit of a stimulant for enzyme release. And so we refer to the fact that these two stimulants are kind of in synergy, right? Like they activate each other a little bit as potentiation. So we say that CCK potentiates secretin's effect. So like as it's stimulating bicarbonate release a little bit, it makes secretin work better. So CCK is potentiating secretin. And so therefore it does have an effect on bicarbonate release, even though that's not like its main job. And so for secretin is potentiating CCK. So it makes CCK like work better. It's stimulating a little bit of enzyme release. So we say that secretin potentiates CCK's effect on enzyme release. Okay. So each of these stimuli that we're making basically have stimuli that release them, and they're kind of what you would think. So CCK's main job, sorry, scrolling back for a second. CCK's main job, this big bullet point, is to stimulate acinar cells to secrete enzymes. The reason we want to secrete enzymes is to break down fats and continue potentially breaking down some proteins. So if we sense fat, if we sense some amino acids in the duodenum, that means we want enzymes to break them down. So that's the stimulus for CCK release. Now, secretin's job, scrolling back again, secretin's job is to stimulate the duct cells to secrete bicarbonate. So secretin's job is basically to help us buffer out acidity. That's why we want that bicarbonate. So the stimulus for the release of secretin is acidity in the duodenum. So if we sense acidity, we know that we want to buffer it out. So we need secretin to tell our duct cells to, we need secretin to tell our duct cells to make bicarbonate. So we could have flowcharts for this. So this one is linking together, this one's basically talking about secretin. Okay, so what's happening here is that when we sense acidity in the duodenum, and there are reasons we sense acidity in the duodenum, right, is we're sending chyme from the stomach. Your stomach made a bunch of acid, so that acid, as we squeeze food and chyme into the duodenum, is coming along with it. So this means that the food and the acid have arrived in our duodenum, in our small intestine. The job of the duodenum is partially to, it's doing its two things, right? It's continuing digestion, but we also need to neutralize that acidity. You don't want to like poop out a bunch of acid, right? It would be unpleasant. So in your duodenum, we have chemoreceptors. A chemoreceptor is always something that's detecting and responding to some type of chemical. So the chemoreceptors in the duodenum are detecting and responding to basically those hydrogen ions, responding to acid. And we have some nervous pathways. So we have short reflexes, we have long reflexes. Both of them affect endocrine cells. Endocrine cells are cells that put a signal into the bloodstream. In this case, our response is to acid. So what we want to secrete is secretin. So our endocrine cells, when we sense acid, release secretin. Secretin gets into your plasma. And then from the plasma, it gets to our pancreatic duct cells because we have blood flow to the pancreas. So the pancreatic duct cells now sense that there's secretin there. Secretin turns on the release of fluid. The main functional component of that fluid is bicarbonate. Bicarbonate then moves through the duct. It moves all the way down to the sphincter of Oddi, not listed on this diagram, but that's the sphincter that allows the release of stuff from our liver, gallbladder, and pancreas into the duodenum. So that bicarbonate-rich fluid going through our duct, collecting more and more bicarbonate, eventually dumped into the duodenum. Bicarbonate, it's like point in a lot of physiology, is to bind on to hydrogen ions and therefore to buffer out acidity. So in the duodenum, when we get that bicarbonate-rich fluid, it's binding onto those hydrogen ions, it's neutralizing our acids. Since it's like removing hydrogen ions from this fluid in the duodenum, total acidity of the duodenum goes down. So that's why we have this down arrow on acid in the duodenum. Now we don't have very much acid in the duodenum, so this whole diagram flips, right? So now that we don't have any acid in the duodenum, this is a negative feedback loop, so it's turning everything off. So once we don't have any acid in the duodenum, can our chemoreceptors respond to acid? No, can't respond to acid that's not there. So our chemoreceptors stop sensing acid, which means they stop sending signals, both long and short, saying, hey, 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 there's acid. Now they're silent. They're saying, nope, no acid. Which means that our endocrine cells, which before were making a bunch of secretin in response to these signals from the chemoreceptors, they're not going to make secretin anymore. They're not making secretin because there's no signal coming to them. We don't have, right? We don't have our action potentials arriving down those nerves, which is ultimately what turned on those endocrine cells. So we don't have secretin being made by the endocrine cells, which means our plasma levels of secretin are going down, 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 down. We're getting less and less plasma in the, we're getting less and less secretin in the plasma, which means that there's less and less secretin reaching our pancreatic duct cells, which means that our pancreatic duct cells are making less and less bicarbonate, which means that we're neutralizing less and less acid. But that's good, that's helpful, because the whole reason that we turned off this loop was that there wasn't acid in the duodenum anymore. So the difference with the negative feedback loop is that, like, unless you have more acid coming from the stomach, this isn't going to go up anymore. You probably will have more acid arriving from the stomach because the stomach is trying to give you, I'm not going to say a steady flow, but it's trying to get a sort of gradual flow from the stomach. So unless you're at the end of processing all the food in the stomach, probably there'll be like a new loop as you get a new little burst of food coming in. But turning off this pathway doesn't in and of itself increase the acid. Okay. So the other place that we're going to see effects of secretin, or we're going to see the effects of secretin and CCK on other parts of our digestive system, basically, so other glands. But they're just the glands that combine with the pancreatic duct to that sphincter of Oddi to dump into the duodenum. So the way the anatomy works, right, is we had our pancreatic duct, we had our common bile duct joining together to make that sphincter of Oddi, right? So the common bile duct went to the liver, went to the gallbladder. So the two other things that secretin and CCK are regulating are the liver and the gallbladder. So secretin, whose main job on the pancreas was to tell it to make a bunch of bicarbonate, is going to tell the liver to make a bunch of bile. Remember that bile has like our bile salts in it, right? We need those bile salts to break up our lipids so that the lipases can reach the outside. So secretin's going to do that. So secretin's kind of doing two things, right? Bicarbonate, bile secretion. CCK also has a job. CCK's job is not on the liver. CCK is affecting the gallbladder and the sphincter that is releasing these juices into the duodenum. So our gallbladder is like our reservoir of extra bile. So if we were making bile from the liver before, but the sphincter of Oddi was closed, means we would have bile spilling over into the gallbladder because it's just a reservoir. So as secretin is making the liver make more bile, CCK is like squeezing the gallbladder to get the bile that we already made into the juice we're sending into the duodenum, because we want that bile to break up lipids. Okay. CCK is also really the ultimate thing that's letting us get any of these juices into the duodenum in the first place, because there's a sphincter closing off our ducts, right? They can't get into the duodenum until the sphincter of Oddi opens. So CCK is what actually relaxes that sphincter. So that's its other job. So CCK is making enzymes in the pancreas. It's contracting our gallbladder so that we can get bile into the duodenum, so that we can break down our fats. And it's stimulating the relaxation of the sphincter so that this stuff that we're making can actually get into the duodenum. So like if we were to not have CCK, we basically wouldn't be able to put any of this stuff into the duodenum. Not the enzymes, not the bile, but also not the bicarbonate-rich fluid, because even though that's made by secretin, if it can't get into the duodenum, it's not there, it can't do its job. So here is a big diagram of how all of this stuff is working together. So in the duodenum, as we have chyme coming in from the stomach, we're doing a couple of things. Part of the chyme is our stomach acids. Part of the chyme is the stuff that we're digesting. So specifically, the stuff that we're digesting that your duodenum is sensing is like proteins and fat. Okay. So acids in the duodenum turn on secretin. Secretin does a couple things. One of the things secretin does is make bicarb. And the other thing it does is make bile. So as it makes bile, bile eventually is going to get into the duodenum. The bile is going to help us neutralize acid. So this fluid that we're all putting together has bicarb in it, and it has bile salts. So the bile salts emulsify fats. The bicarb neutralizes acids. On the protein digestion and fat side, as we sense proteins and fat in the duodenum, this turns on this pathway where we have this signal CCK. This signal CCK tells the gallbladder to contract, so that's squeezing bile into the duodenum. The other thing it does is relax our sphincter going into the duodenum, which means that all of this stuff can get into the duodenum. So that is also going to help us neutralize acids and emulsify fats. Okay. We're gonna come back to this in a second. Hopefully we'll have time to like work through at least one of these questions together so that we can kind of practice for our last exam. But before that, I want to just make sure that we get to peristalsis and segmentation. So peristalsis and segmentation is the motility part of gastrointestinal, regulation. So motility just means movement. So when we say there's GI motility, we mean that like muscles in the wall of the GI tract. Remember that there are muscles in two different places. One's in our mucosa layer. So in that first layer, we have some muscle, some circular muscle, some longitudinal muscle. We also have an outer layer of muscle called the muscularis externa. So this is a bigger layer of muscle. The bigger layer of muscle is the one that does most of GI motility because it's bigger. The point of GI motility is to mix around the stuff in our GI tract. The reason we want to mix around the stuff in our GI tract is so that all the food we've eaten actually contacts the cells lining the GI tract, because if those amino acids are like in the center of the lumen, they're just going through, right? Like you can't get absorbed by a cell that you're not touching. So we're trying to squish around all the food so that it touches the cells lining the tract and can get absorbed, because once you touch the membrane, then you could potentially get absorbed. And the other thing is we do want to keep moving our like waste products through. So we're also going to try to propel the waste products down towards the end of the system so it can be excreted as feces if we don't need it. This basically happens through like waves of contraction. Okay, so we have two basic terms when we're thinking about GI motility. We have peristalsis and we have segmentation. So peristalsis is basically the waves that are moving the waste products forward. So as we're like squeezing stuff like a tube of toothpaste down through the GI tract, that's peristalsis. And this is happening basically in response to slow waves moving through the nervous system in the GI tract. So like action potentials that are being produced by the cells in our GI tract have this electrical rhythm that we call the basic electrical rhythm. So the basic electrical rhythm is just a wave of contraction that's kind of naturally there in the GI tract. Okay. So one of the things that this natural basic electrical rhythm is causing is peristalsis, moving waste products forward. The other thing it's causing is segmentation. And segmentation is the part where we're just trying to like jostle all the food so that it'll contact the walls of the GI tract so that we can absorb the stuff. Okay. So if we wanted to look at a diagram of peristalsis, it would look something like this. So over here in this image, right at the top, we have the proximal part of the GI tract, and at the bottom, we have like more distal parts. So what's happening in peristalsis is we're just trying to move like our ball of food or ball of chyme or ball of whatever down through the GI tract. And we're gonna need to use both our circular muscles and our longitudinal muscle to do this. And we're gonna like basically be using the part more proximal to our ball of food as well as the part more distal to our ball of food. So we're trying to squeeze the food forward. So we're squeezing it kind of like a tube of toothpaste. So we're gonna be trying to narrow the part where the food was and behind it so that it doesn't move backwards. The other thing we're doing is trying to make like the next part of the GI tract where we're moving our waste products to, we're trying to make it wider, right? We're trying to make it easier for the food to actually get there. So we're gonna be like trying to increase the diameter there. The way we're doing that is like actually by increasing the circular diameter as well as like kind of making it shorter. So behind our ball of food, proximal to it, we're trying to make it super narrow. And so two ways we can make it super narrow are by contracting circular muscle, so actually closing down the diameter. And when we relax longitudinal muscle, it also makes that like a little longer. So if it's long and narrow, it's like creating a real block. So it'd be hard for the food to move backwards. And also ahead of the food, in our distal segment, our circular muscle is relaxing and our longitudinal muscle is contracting. So longitudinal muscle is squishing this up. So it's squishing it up. So we have like a lot of room. This is getting very short. Our food has a lot of room here. And then as that ball of food moves forward, we then have the wave continue. So this distal segment would be where the food is now. So it would now become the proximal segment for the next phase of peristalsis. So this all really just propels the food forward in a way that, if you think it through, is hopefully like relatively like what you would expect, right? As you move food, you're moving it from a small space to where there's more space. You're just kind of squeezing it. And segmentation is happening in the small intestine specifically. So the point of segmentation is for us to be able to absorb nutrients, and the place that nutrients get absorbed is the small intestine. So segmentation is really mainly happening in response to the circular muscle. So before we were using both the circular muscle and the longitudinal muscle. Now we're really mainly thinking about circular muscle. And what's basically happening is that like when you looked at the intestine, you saw that there are like kind of little bubbles, right? There are little segments. It's kind of lumpy, okay? So we think about those as different segments of our intestine. And we're basically trying to push some of the food, like kind of jostle it backwards and forwards. So the way we do that is just like squeezing alternate segments, and it just kind of mixes it all up. So that's it. It's that throughout our intestinal system, we're trying to squeeze this ball of food forward, but in our small intestine, we're also kind of mixing it around. So the circular muscle is doing a little bit of a different thing if we're in the small intestine. Okay. I don't want to stop share. Maybe I do. Oh no, I just want to exit this. I don't know what that is. So I want to try, how should we do this? I want to try working through one of these checkpoint questions so that we get a little practice. Which one looks good? Let's start with just the first one since it's first. So on Friday, we talked a bit about acid secretion so like the beginning of acid secretion and the actual cells. So a question we would hopefully be able to answer on that is something about how the acid is actually getting released. So one of the things happening is that we're going to be using carbonic anhydrase. So I want you to take a second and see if you can find where we have carbonic anhydrase anywhere in this process. I think I have a picture in my slides, but I'm not sure that you do in your slides. So see if we can find the figure that's going to show how this influences stomach acid secretion, or if you have in your notes written how this works. Take a second to answer this question, and then I'm going to kind of show you how I would go about answering that question. So I'm going to be quiet for a couple minutes and let you do that. Okay. So did anyone find a point in their notes or a picture that would help them answer this question? And you can pop an answer in the chat or turn on your microphone. Either works. All right, I'm gonna take that as a no. So what we would have been looking for, I'm gonna share my other screen. Okay, screen 2. Okay. So one of the figures in your textbook that I popped into my set of the slides is this figure. figure 20.24. So the way we would start to figure out how to answer this question, right, is we're looking for stomach acid secretion. We're talking about carbonic anhydrase, which hopefully maybe you kind of remember is part of how we make hydrogen ions and bicarbonate ions. So what we're looking for is like a picture of the actual cells that make these things. So we're looking for a picture of cells in the stomach itself. So here we have a picture of a parietal cell. So a parietal cell is what's making this acid. So here's a picture of it. And the part that's carbonic anhydrase, here's the Ca. And it doesn't, actually, does it tell you here? I'm not sure it tells you that in the figure. Okay. But it's used that symbol before for carbonic anhydrase when we're talking about pH. So here, the role of carbonic anhydrase that we would be looking for is that carbonic anhydrase is what helps us take CO2 and water together to make carbonic acid, which breaks into hydrogen ions and bicarbonate. Okay, so that would be the first part of the answer here. So one of the ways that carbonic anhydrase is helping with stomach acid secretion is that it's what's actually basically making our hydrogen ions. The second part of this question is asking us about what else, what other ions are getting secreted with our hydrogen ions. So basically, we would look at this side of the cell and see what's going into the lumen with our hydrogen ions. So nothing's getting directly, like as in with the same transporter, moved out of the cell with the hydrogen ions, but we can see two other things. We have our chloride ions and our potassium ions are also at this point getting secreted into the lumen. So we might want to try to figure out why. And so let's focus on the chloride ions first. So one of the things that happened here when we made our hydrogen ions is we also made bicarbonate ions. We have to put bicarbonate ions into the plasma. So we're putting them into the plasma. And this is happening basically through an antiporter. So we're having chloride ions move in as we move the bicarbonate out. So then we have these chloride ions in the cell. They're kind of attracted to the positive charge in the hydrogen. And so they move out with it through their own basically channel into the lumen. And then the other part of this is the potassium. So potassium is being moved into the cell directly as the hydrogen is moving out. And this is creating kind of a gradient of the potassium. So as we're moving hydrogen out, we're building up a high potassium gradient inside our cell. There's a channel that releases that potassium back into the lumen so that this can kind of happen as a loop. So potassium is also being secreted. So that's kind of how we would break down this question. It's like, look for a figure that basically explains it, or at least I like figures and pictures best. And then try to break down those parts. Okay. I think, yeah, we're gonna finish. I know I'm over time a little. Okay. So possibly expect another update on something from me this afternoon, but that's it for now.